Hello everyone and welcome to Samiki. My name is Giovanna Prado. I am a biotechnology student here at PUC-PR, and in this past year I was advised by Professor Vanessa Santos Sotomayor in our project named as rs2023004 Alleles from PARK2 gene and Parkin expression in colorectal adenocarcinoma. So colorectal cancer is a type of malignant neoplasm that affects the colon and the rectum. For the year 2020, it was the third most incident type of cancer in the world. The tumor behavior is highly dependent on the genetic and epigenetic changes it accumulates, which can be different between patients. Therefore, it is important to characterize tumors by their genetic mutations to give the correct prognosis to a patient. In 2018, Da Silva Camargo et al. Saw that, among 73 cases of colorectal cancer, those who had higher Parkin expression in profound tumor areas had higher survival rates. Although this discovery already has the potential to help health professionals with patient prognosis, the article doesn't clarify why we see this variation in Parkin's expression between patients. There are some reasons this could happen, but here we will explore further the possibility of a single nucleotide polymorphism, more specifically rs2023004, being the cause of this variation. We use the same 73 samples that Da Silva Camargo et al. Used in her article. We also had samples from tumor borders from those same patients and some control samples from patients who had the surgery for other reasons. We first had to extract the DNA out of the purifying samples, which is a total 2-day protocol. After this, we could start with the allelic discrimination process. We used QuantiStudio from Thermo Fisher for this. Lastly, we did a statistical analysis to compare the average expression from each genotype of rs2023004 with one another. We used ANOVA and student t-test for this. So here are the results for the tumor samples. We can see that most of them are heterozygous CT and the minority of them are homozygous CC. We can see the same thing for the surgical border samples and kind of the same thing for the control samples, but we needed more samples to say that. So yeah. Here are the results for the statistical analysis. So we can see here that all p-values are above 5%. Here we can see this as well, which means that all genotypes are statistically equal. So therefore, we can conclude that rs2023004 genotypes have no influence on the expression of Parkin in the analyzed samples. However, this polymorphism has been associated with higher susceptibility to leprosy. So it is possible that this SNP, while it doesn't affect the protein expression, affects its function. We could test this by comparing the expression of Parkin's target proteins, such as BCL2L1, to the rs2023004 different genotypes. So I have a lot of people to acknowledge here. Firstly, Professor Vanessa Santos Sotomayor, thank you very much. Pookie, also thank you very much for this opportunity. And everyone from Lab 1, you guys have my heart. Thank you for everything.